Hello friends, warm welcome in this video, that is the antigenicity and immunogenicity. Friends, before understanding the concept of antigenicity or immunogenicity, we should know the meaning of antigen. So an antigen is any substance that can be recognized by antibody or T cell receptor. The antibodies are the membrane receptor present on B cells, or it may secreted into the serum by the B cell. In the similar fashion, the T cell receptors are present on the T cell. Although both antibody and T cell receptor recognizes antigen, but their recognition pattern is somewhat different. Antibody have a capability to directly recognize the antigen, but the T cell receptor can't recognize antigens until and unless it is complex with the MSC molecules which are present on antigen presenting cells. Once the antigen is processed and presented by antigen presenting cells, only then the T cell receptors can recognize them. So what is immunogenicity? Immunogenicity is the ability of a foreign particle or a antigen to induce humoral and cellular immunity. Humoral immunity is the immunity which is provided by antibodies, and the cellular immunity is the immunity provided by T and related cells. It means that if a immune response is evoked by a foreign particle, then this phenomena is called as immunogenicity. For example, when naive B cells, that means the B cells which are not previously encountered with any antigen, encounters the antigen that matches its membrane bound antibody. The binding of antigen to the antibody causes the B cell to divide rapidly and induce immune response. The progeny of rapidly divided cells differentiated into memory B cells and effector B cells, that is called as plasma cells. The memory B cells have a longer lifespan than the naive cells, and they express the same membrane bound antibody as their parent B cell. On the contrary, plasma cells produce the antibody in a form that can be secreted and have little or no membrane bound antibody. Although plasma cells live for only few days, they secrete enormous amount of antibody during this time. It has been estimated that a single plasma cell can secrete more than 2000 molecules of antibody per second. Secreted antibodies are the major effector molecules of humoral immunity. In the similar fashion, when naive T cells combines with antigen, they induce an immune response and produce effector T cells and memory T cells. The effector T cells include cytotoxic T lymphocytes and T helper cells. Thus, any substance that induce immune response is called as antigen, but the more appropriate term is immunogen. On the contrary, antigenicity is the ability to combine with the final product generated during the immune response. For example, the final product generated during the immune response by B cell is antibody. When these antibodies interacts with antigen, they form antigen-antibody complex. This property of antigen to combine with the antibody is termed as antigenicity. In the similar fashion, when a T cell receptor combines with an antigen to form antigen T cell complex, it is termed as antigenicity. So antigen is any molecule which have a capability to combine with T cell or B cell receptors. No doubt, immunogenicity and antigenicity are related terms, but they are distinct in their immunologic properties. All molecules that have the property of immunogenicity also have the property of antigenicity, but the vice versa is not true. Some small molecules like haptens are antigenic but not immunogenic. Means that they have capability to combine with antibodies or T cell receptor, but they can't induce the specific immune response themselves. But when these haptens combines with some carrier proteins, they form hapten carrier conjugate, which have a property to induce a immune response. So friends, this is the difference between immunogenicity and antigenicity. If you like this video, please subscribe to our channel, press the bell icon, and don't forget to share this video with your friends. Thank you.